Hello guys, welcome to our today's lesson, and we want to talk about the plant cell and animal cell as seen under an electron microscope. Previously we saw the plant cell and animal cell as seen under a light microscope, and the cell structures, which we call them the cell organelles, were very few. Basically, the cell membrane, the nucleus, and the cytoplasm are the ones which were seen. But in our case, under the electron microscope, we are able to see more structures. We are able to see more structure with the more cell organelles. For example, the nucleus, rough endoplasmic reticulum, cell membrane, mitochondrion here, the lysosomes, the Golgi apparatus. Those are the sum of the cell structure of organelles which are seen under an electron microscope, simply because it has a more magnification power than a light microscope. And as you can see here, these are an animal cell, simply because it only has the cell membrane. But later on we shall look at the plant cell and animal cell. But in the plant cell, the outermost organelle or the structure is a cell wall. The plant cell have cell wall, but the animal cell have only the cell membrane. We can look at the two structures, the cells. Here we have the animal cell, and on my right we have the plant cell. You can see the difference. The animal cell has this cell membrane only, which is the outermost layer, while in plant cell the outermost layer is a cell wall. Then after the cell wall we have the cell membrane. That is the main difference between the plant cell and animal cell. And also another difference, the plant cell has a chloroplast, a cell organelle called chloroplast, which is able to trap light energy and manufacture food, while this animal cell lacks the chloroplast. Back to our cell structures or the cell organelles. As you can see, you are able to see more cell organelles or cell structures under an electron microscope, like the smooth endoplasmic reticulum, nuclear pore, chromatin, nucleolus, mitochondria, Golgi apparatus, name them. And these cell structures, we shall look at their functions and how they are made up, or their structure. Next, what is a cell? You can ask yourself, what is a cell? And a cell is a basic unit of structure and function in the organism. This means our whole body is made up of very many cells. Later on we shall look at it. Very many cells make up tissues, and the tissues make up organ make up organ system, and the organ system is able to make up our body. And most of these cells, they are very small and they are invisible. That is, when you are not using a microscope, you cannot be able to see these cells. Next, let's look at the cell theory. And the cell theory says that every living organism is composed of cells, and every cell in an organism is produced by another cell, and that is through cell division. We have two types of cell division. That is mitosis, the cells dividing to form more cells. That is the body cells or the somatic cells. That means that we must have a cell which will divide to produce other cells. Then maybe the reproductive cells are gotten through cell division by meiosis. That is another topic We shall talk about it later on. Let's look at the cell structure. And the cell structure contains what we call cell organelles. They contain what we call the cell organelles. And these cell organelles are basically these structures found within the cell. For example, the nucleus, the cell membrane, the mitochondrion, the lysosomes, the Golgi apparatus. We'll talk about these cell organelles in this lesson. As you can see, in the examples of organelles we can have the lysosomes here. This is the chloroplast, it is green. Golgi apparatus. You have mitochondrion, endoplasmic reticula. Can have the nucleus here, the ribosome here. Those are examples of the cell organelles. We also have our cell here. This is the animal cell here, which contains those cell organelles. And apart from cell organelles, we have what we call cell inclusions, which can include starch droplets, lipid droplets. Those are some of the cell inclusions. And the organelles are structures that enable the cell to live, grow, and reproduce. Let's start with the cell membrane. And the cell membrane, in this case, it is the outermost structure of the cell, and that is the animal cell. In the animal cell, it is the outermost structure. While in plant cell, the outermost structure is the cell wall, and from the cell wall we have the cell membrane. But in this case, these cells we are looking at, they are animal cell, and this is the outermost layer, is what we call the animal cell membrane, as you can see. And the other name for cell membrane is a plasma membrane. The structure of the cell membrane, how does it look like, or what does it entail or contain? It contains 3 layers when seen under an electron microscope. We have 1 phospholipid layer and 2 protein layers. As you can see here, let me take you here. We have this protein layer here, another protein layer, then phospholipid layer sandwiched between the 2 protein layers. And these protein layers and the phospholipid layer are very important because they'll affect the property of the cell membrane. For example, the cell membrane has certain properties. It is sensitive to changes in temperature and pH. That is due to the protein layer. And because the proteins are affected by changes in temperature and pH. Then let's look at the role of a cell membrane. What is the role of the cell membrane? Being the outermost layer in the animal cell, it selectively controls movement of materials into and out of the cell. Selectively means materials which have large size or large molecular size cannot pass through. Only materials with a smaller or small molecular size will pass through into the cell. That's why we say it is selectively permeable. Then also contains or encloses cell contents. It encloses the cell contents. As you can see here, the contents are here inside. It encloses them. Then you can describe this cell membrane as a gate into the city. As you can see, the gate into the city selectively allows certain goods or people to enter into the city. That's why we compare it with a gate into the city. Back to the structure, we said it has two protein layers and a phospholipid layer sandwiched or in between the two protein layers. Then the other view can see the proteins here and the protein here. Then we have a phospholipid layer sandwiched, and we have what you call pores, and these pores allow materials in and out of the cell. And these pores, they say, they are small size, only to allow molecules which have a small size. Let's look at the cytoplasm. Cytoplasm is a jelly-like fluid contained in the cell, and it holds the cell organelles. In short, we are saying this cytoplasm is everything within the cell membrane which is not in the nucleus, is what we call cytoplasm. And this cytoplasm makes up most of the mass of many cells. So basically, this jelly-like fluid here, you're seeing on this arrow, the jelly-like fluid is what we call the cytoplasm. And the role is this is where chemical reactions occur. Remember, we have several chemical reactions which occur within the cell, and they occur within the fluid medium, which is the cytoplasm. Apart from that, it contains other organelles and dissolved substances, for example, glycogen and starch droplets. Then this cytoplasm entails cytosol, which is a jelly-like mixture in which other organelles are suspended. So the cytosol plus organelles, we get the cytoplasm. So basically, the cytosol is a jelly-like mixture where the organelles are suspended. When you add up the two, you get what you call the cytoplasm. And this cytoplasm, remember, it is not static, but it has a movement called cytoplasmic streaming. It has a movement called the cytoplasmic streaming. And this cytoplasmic streaming is important because it helps distribute materials and cell organelles within the cell. That is the importance of the cytoplasmic streaming, or the movement of cytoplasm within the cell. Let's look at the nucleus. And the nucleus is round in shape or oval, as you can see here. It is round in shape or oval, and it is near the center, that is mainly in an animal cell. And it is separated from the cytoplasm by a thin membrane. We call it nuclear membrane. Nuclear membrane. You can see the structure of the nucleus. You can see here the nucleus. We have our nucleus. There at the center, this is the nucleus. Then we have enlarged it. As you can see, the nucleus has what we call a nucleolus, which is responsible for synthesis of ribosomes. Then it has chromatin here. Chromatin are hereditary materials. Then nucleoplasm. A nucleoplasm, the word plasm, means a fluid found in the nucleus. Then the outer membrane. Then we have pores which allow materials in and out of the nucleus. Let's look at the role of a nucleus. What is the role of the nucleus? It controls cell activities. It means when the nucleus is dead, the cell will be dead, because there will be no control of cell activities, thus leading to death of the cell. So the role of the nucleus controls cell activities, which can include protein synthesis, cell division. and it contains, as I've said, it contains the nuclear membrane, and the nuclear membrane has a nuclear pore which allows and controls movement of substances in and out of the nucleus. You can have the nuclear membrane here. This is the nuclear membrane. Then we have the nuclear pore here, another one here, which allows materials in and out of the nucleus. Then the nucleoplasm, these are fluid which has a nucleolus and chromatin, and the nucleoplasm is found inside the nucleus. The nucleolus manufactures ribosomes, and the ribosomes are site for protein synthesis. We shall talk about it later on. Then chromatin contains hereditary or genetic materials which enable the characteristics of a living organism to be transferred from the parent to the offspring. That is a chromatin. We have an animal cell showing is a nucleus. You can see the nucleolus here, then nuclear membrane here, then we have the nucleus itself. This is the nucleus itself. Then we have some pores here which enable movement of materials in and out of the cell. Then this fluid here, we have said it is called nucleoplasm. We have another view of the nucleus. At the center here, this is the nucleolus. Then we have this the nuclear membrane. Then we have these pores. This is the nuclear pore, which we said controls movement of materials in and out of the nucleus. Then we have nucleoplasm. This is the fluid inside the nucleus which contains this nucleolus and the chromatin or hereditary materials. Next organelle, we have mitochondria, and the mitochondria, it is rod-shaped. It is a rod-shaped. Let me take you to the shape of the mitochondria. As you can see, it is like a rod. It is rod-shaped. Also, we have these other structures, other mitochondrion here. It is rod-shaped, and the inner membrane is greatly folded into what we call crista, and the crista are just finger-like projections, and they increase surface area for attachment of respiratory enzymes. Remember, in the mitochondria, that is where respiration occurs and yields energy for the cell. And in this case, these finger-like projections, we call them crista. They increase surface area for attachment of respiratory enzymes which catalyze respiration reactions. Then has a matrix, and the matrix is the fluid found inside the mitochondrion, and it provides a site for respiration. And a point to note, actively or rapidly respiring cells, for example, muscle cells, sperm cell, apical meristems, kidney cells, have numerous mitochondria, and that is to provide energy for the functioning of these cells because they require more energy. Role of the mitochondria, and that is provides a site for respiration to yield energy for the cell, and basically we refer to them as powerhouse of the cell due to production of that energy. As you can see, we have this matrix. Matrix is the fluid part where respiratory reactions occur. Then crista, you can see their inner or contains inner membrane which is highly folded into finger-like projections to increase surface area for attachment of respiratory enzymes that speed up or catalyze respiratory reactions. Then we have the outer membrane here. This is just a repetition, outer membrane. Then we have the many inner projections, crista, which increase surface area. Next we have ribosomes. Ribosomes are spherical in shape, as you can see here, and they're scattered within the cytoplasm, and while others they are studded or embedded on the surface of the rough endoplasmic reticulum. And what is the role? The role is they provide a site for protein synthesis. Next, let's look at endoplasmic reticulum. And the endoplasmic reticulum contained interconnected channels which are continuous with the outer membrane of the nuclear membrane, and they include smooth endoplasmic reticulum and rough endoplasmic reticulum. Why smooth? Because they lack ribosomes on their surface, while rough endoplasmic reticulum, they have ribosomes on their surface. As we have said earlier on about the ribosomes, are found within the cell, and others are embedded onto the rough endoplasmic reticulum. And basically, this endoplasmic reticulum, can describe them as roadways of the cell. Roadways, it means pathways of the cell. It provides a path or road to transport cell secretions like proteins and lipids. Let's look at the role of the rough endoplasmic reticulum, and it transports proteins, while the smooth endoplasmic reticulum manufactures or synthesizes and transports lipids. Let's look at these diagrams. The first place we have rough endoplasmic reticulum. As you can see, they're just made up of interconnected channels containing ribosomes on their surface. The ribosomes provide surface or sites for protein synthesis, and these channels transport proteins. You can see the ribosomes, very many ribosomes on the rough endoplasmic reticulum. This other view, the interconnected channels, we call them cisternae, which are able to transport the proteins produced by ribosomes. The smooth endoplasmic reticulum, that one transports lipids. It does not have ribosomes on its surface, but the cisternae, it is just interconnected channels. Then on this other hand, we have rough endoplasmic reticulum. Their surface made up of very many ribosomes. The ribosomes produce proteins, which are transported by the rough endoplasmic reticulum. Cisternae, as I've said, consists of interconnected channels. Also, we have another view. We have this view. As you can see, we say that this endoplasmic reticula are in connection with the nuclear membrane here. We have the smooth endoplasmic reticulum here. Then on the other hand, we have rough endoplasmic reticulum that are in connection or continuous with the nuclear membrane of the nucleus here. And the role, rough endoplasmic, I've said, transports proteins, while smooth endoplasmic reticulum transports lipids. Next, we have what we call Golgi bodies, or in other words, we call them Golgi apparatus, and contains a series of flattened sacs, and they are stacked of membrane-bound tube-like sacs and found close to the cell membrane. Let's look at those sacs we are talking about. You can see the Golgi apparatus, and they have vesicles, and they look like sacs. As you can see these diagrams, just looking like sacs. Their role is to package and transport glycoproteins, and the glycoproteins is made up of glycogen and proteins and carbohydrates. And also they transport synthesized materials or substances out of the cell as secretions, e.g. enzymes. They are transported by Golgi bodies. Apart from that, secretion of synthesized or manufactured glycoproteins, that is also their role, and also manufactures lysosomes. Lysosomes, we'll talk about it later on. And NB, a point note, materials that are transported by endoplasmic reticulum usually stop first at the Golgi bodies, where they are stored or altered before moving to other parts of the cell. That is a great point to note. The materials transported by endoplasmic reticulum, for example, the proteins and the lipids, they are stored first in the Golgi apparatus, and the Golgi apparatus will package them into glycoproteins and transport them to other parts of the cell. Let's look at the Golgi apparatus. It looks like this. It contains Golgi vesicles, which contain secretory granules for cell secretions. Then the vesicles, this is how the Golgi apparatus looks like. Then we have a three-dimension drawing showing a Golgi body in action. As you can see, the secretory vesicles of the Golgi apparatus. Here, the Golgi apparatus itself. Then on the other hand, we have a rough endoplasmic reticulum. And the rough endoplasmic reticulum will transport the proteins of the cell secretions to the Golgi apparatus. The Golgi apparatus will store those proteins and modifies them into glycoproteins, then will transport to the cells where they are required. That is how the Golgi apparatus works. Next we have lysosomes. And lysosomes, they are round in shape, and their role is that it contains lytic enzymes. Keyword is lytic enzymes, which destroy worn out cell organelles and also digest big food materials or break down food materials. And when setting the function, as I've said, you must say they contain lytic enzymes. And this is the structure of the lysosome here. And a point to note, white blood cells contain numerous lysosomes because they destroy pathogens, hence contain numerous lysosomes because of they contain lysosomes which contain lytic enzymes that destroy pathogens. As you can see, the lysosomes having vesicles which contain lytic enzymes that destroy worn out cell organelles. Next we have vacuoles, and the vacuoles are sacs which are filled with fluid. For example, these are plant cell, we have the sap vacuole, but in animal cell they are very small. As you can see, they are just sacs which contain fluid and vary in size. And animal cells contain small vacuoles which are small and temporary, while the plant cells, they have large and permanent vacuoles. As you can see, these are sap vacuole which is large and permanent, while in animal cell they are very small. They contain in plants, vacuoles are centrally placed and surrounded by a membrane called tonoplast and a solution of salt and sugars called cell sap, hence they are called sap vacuoles. Their roles is to store salt and sugar, and the sap contributes to osmotic properties of the cell. Then in unicellular organisms, we have what we call contractile vacuole. And the contractile vacuole removes or excretes excess water through osmoregulation. Then also excretes metabolic waste. Another type of vacuole in unicellular organisms, we have food vacuole, and it is for feeding or digestion or storage of food. You must know all those types of vacuoles. And these unicellular organisms, mainly we have amoeba, paramecium, and we're saying they have or they are single celled, only having one cell. So the vacuoles, you must know all of them, and the plant cell, they are very important because they are large and centrally placed, and they contain the salts and sugars which contribute to the osmotic properties of the cell. As you can see, this vacuole in the plant cell here, very large at the center, stores water, food and waste. Then it is the largest in plant cell, and as you can see, when these vacuoles are filled with water, the leaves will become, or the cells become turgid, and erect, while when the vacuoles have less amounts of water, the plants will droop. So it is important in storage of water and those salts, thus affecting osmotic pressure of the plant cells. Then next we have cell wall. Cell wall only found in plants. The animals lack a cell wall, and this cell wall, apart from being found in plants, it also found in fungi and in some prototists, but not in animal cells. And it is made up of cellulose that is in plant cell and either chitin or cellulose in fungi. And it is very rigid and tough. The role protects the cell against mechanical injury, and it gives the cell a definite shape. That is why these plant cells, as you can see, they have a definite shape. You can see the definite shape here. It does not change. Then also provides mechanical support due to the presence of cellulose. Next we have chloroplasts, and the chloroplasts are only found in plant cells. They contain chlorophyll, and this green coloring matter is what we call the chlorophyll. They make the plant green, and the role of chloroplasts, because they contain chlorophyll which traps light energy for photosynthesis. Thus the chloroplast provides a site for protein synthesis. You can look at the structure of chloroplast. You can see the inner membrane, outer membrane. Then we have the stroma, where carbon dioxide fixation takes place. That is during the light independent or dark stage. Then we have the grana. These stacks, call them the grana, which contains chlorophyll, traps light energy for splitting of water molecules, or what we call photolysis. That is during light stage. That is the chloroplast. Then we have centrioles, and the centrioles, they are rod shaped, as you can see, like rods, are located just outside the nuclear membrane, and they form spindle fibers during cell division in animal cells. Then apart from that, they help form cilia and flagella in cells, basically most unicellular cells or organisms, for example under kingdom Protoctista. And these cilia and flagella are used for movement. Then we have two cell types. We have prokaryotic cell, which is a very simple, and a prokaryotic, it is not membrane bound, or the structures are not membrane bound. Common example is bacteria under kingdom Monera. While the eukaryotic cell, they are more advanced and has membrane bound structures. For example, the nucleus has a nuclear membrane in the animal cells and the plant cells. You can see the example of a prokaryotic cell and a eukaryotic cell. And in this prokaryotic cell, you can see the DNA and the nuclear region, they are not enclosed by a membrane. And on the other hand, the nucleus here contains a nuclear membrane surrounding it. That is the difference between a eukaryotic cell and a prokaryotic cell. The eukaryotic cell contains organelles which are membrane bound, while prokaryotic cell contains organelles which are not membrane bound. Just a repetition, prokaryotic cells have no membrane covered nucleus. It means they lack nuclear membrane, and they have no membrane covered organelles, and their DNA is circular. Examples, we have bacteria. On the other hand, the eukaryotic cells, they have a nucleus, and the nucleus is covered by membrane, also the organelles covered by a membrane. Then the DNA is linear in a line. And the examples, we have those animal cells. Then let's look at the organelles found in Only in plant cells we have the chloroplasts and the cell wall. Then found in plant and animal cells we have the nucleus, Golgi apparatus, mitochondria, lysosomes, endoplasmic reticular, cell membrane, ribosomes, and vacuoles. Then let's look at organization of living things. We have 5 main levels of organization. The first level we talked about, it is cell, basic structure, functional unit of a living organism. And the cells make up the tissue. Many tissues make up organ system, making up organism. Tissue, example, we have muscle tissue. It is made up of specialized cells which come together to perform a specific function. Then organs, for example, we have the heart, the lungs, brains, found in both plants and animals, and composed of mainly tissues that is organized into groups that work together to perform special functions. Then organ system, an example, we have circulatory system, and it is made up of a group of organisms that work together through a coordinated way to enable functioning of living organisms. You can see digestive system, skeletal system, then the circulatory system. 10 organ system in complex organisms can have the circulatory, digestive system, nervous system, respiratory system, skin system, skeletal, that is, skin is also called the integumentary system, muscular system, reproductive system, excretory system, endocrine system, which is hormonal or made up of hormones. Then organisms, we have different organ systems work together to keep the organisms alive. Question, classify the following as a tissue, organ, or organ system. The brain is an organ, spinal cord, organ, nerves are cell tissues. The heart is an organ, group of muscle cells, those are tissues. Question, can a single cell organism contain a tissue? No, because it has only one cell. Give one example of each cell. We can have a sperm cell, organ, the heart, tissue, muscle tissue, organism. Can have a frog. Thank you for listening. Kindly subscribe, like, comment, share, and we'll give more content. The next lesson we'll talk about the microscope and the cell physiology. Thank you for listening.